Enzymes. Plan of the lecture. We discuss now structure of enzymes, how do enzymes work, catalytic mechanism, effects of temperature and pH on enzyme activity. Structure of enzymes. In most cases, enzymes are proteins with the exception of few catalytic RNAs which were found in nucleus. They act as a catalyst compounds that increase the rate of chemical reactions. Many enzymes require cofactors, non-protein compounds that participate in the catalytic process, coenzymes, derivatives of vitamins, metal ions, and metallocoenzymes like heme. The active or catalytic site is a part of the enzyme molecule at which the reaction occurs. The active site binds substrate, the molecule that is acted upon by the enzyme cofactors, prosteatic groups, and contains amino acid residues that helps to hold the substrate and interact with it. Active sites generally occupy approximately less than 5% of total surface area of enzymes, formed by one or more regions of the polypeptide chain. Active site has specific shape due to the tetraeder structure of protein. A change in the shape of protein affects the shape of the active site and the function of enzymes. So different factors like temperature, like pH, or other proteins can change activity of enzymes. These works could be made by small molecules like activators and inhibitors. According to the reactions by the catalyst, enzymes could be classified for several groups. Most enzymes names and in A's. Enzymes usually have both a common name and a systematic classification that includes a name and an enzyme commission number. We can divide all enzymes into 6 groups. Oxide reductase, that transfer of electrons. Transferases, group transfer reactions like amino group or carboxylic group. Hydrolases, they catalyze hydrolysis reactions, transfer of functional group to water. Lyases, addition of groups to double bonds or formation of double bonds by removal of groups. Isomerases, transfer of groups within molecules to yield isometric forms. Ligases, formation of CC and CS-CO-CN bonds by condensation reactions coupled to ATP cleavage to produce ADP. How do enzymes work? Enzymes catalyze reactions by the sequence of formation of enzyme and substrate complex. After that, substrate converted into product, and after that the next step is the releasing of product and freeing of enzyme. The first step, the substrate is a molecule that is connected to the active site and transformed by the enzyme. Enzymes are usually highly specific in terms of their substrates and products, so they can recognize different forms of substrate or different subforms like species of forms like hexoses, like fructose and the glucose. The substrate molecules bind to the substrate binding site, lock and key model, and use-it fit model, and the converted products which are released. The activated substrates and the enzyme form a traditional state complex like enzyme substrate complex at first step and enzyme product complex in the second step of catalyzed reaction. Enzymes use general acid-based catalysis, covalent catalysis and metal ion catalysis, which you already studied in course of chemistry. The main catalytic mechanism of enzymes works like as enzymes decrease the energy of activation for a reaction. They don't affect the equilibrium concentration of the substrates and products, and the number of molecules of enzyme is not changed in these reactions. Effects of temperature and pH on enzyme activity. Enzymes have optimal pH and temperature where they work. The velocity of a reaction increases with temperature until the maximum is reached, after which the velocity decreases with the degeneration of the enzyme. Each enzyme catalysis reaction has an optimal pH also as a temperature, and at which it has maximal activity. At higher or lower pH, activity decreases.